This is the third and final part of the nutrients and their metabolism, and in this one we're going to talk about if we were to do a chemical analysis of a feed, how would we describe the 6 classes of nutrients and get an idea of what amount of each of those classes of nutrients feeds would have in it. So it's looking at the chemical analysis of feeds. The first thing we need to discuss, and we will spend a future activity delving into the different ways of expressing nutrients, but just to provide you a bit of an overview, there's a couple different ways as nutritionists we talk about nutrients. The first is what we call as fed. That's as it's presented to the animal. So if we were to take a feed, it would have a nutrient component and a water component. And all feeds contain water. Even hay or cereal grains that seem fairly dry have about 8 to 10% water in them. Some feeds, such as fresh pasture in the spring, actually have about 80% to 90% water. So it depends on that feed. But that water content can be extremely variable. And as a result, because of that variability, it's something that as nutritionists, especially on animals that are on high moisture diets, we take into account. So when we talk about how a feed is presented to an animal with the water included, that's what we call the as-fed basis. Now, as nutritionists that feed, like especially ruminant nutritionists or equine nutritionists that feed wet feeds that have variable water content, we like to balance rations on what we call a dry matter basis. And that is the basis in which we remove the water and just look at the nutrients. And so we're concentrating the nutrients, but that water, while important to the animal, is acting as a diluent to the other nutrients that we're trying to get into the animal's body. So we express nutrient requirements based upon a dry matter basis. Some species, especially poultry and swine, and even some of your complete feeds that you may buy at a feed store, even dog food and cat food, is expressed on what we call an air dry basis. That represents approximately 90% dry matter, meaning it's not quite 100% dry matter, but it's not complete as fed either. It's an in-between one as well. Most of the time, though, we're going to be working with as fed versus dry matter. One of the things is we take a look at comparing as fed versus dry matter if we're dealing with weight, meaning pounds. Because as fed has the water in it, it's always going to be heavier weight. So if I say a cow requires 20 pounds of dry matter, that's a lighter weight. If we convert it to how much we'd really have to feed her to get that dry material in here, we would need to have a heavier weight because we're adding the water back in. In contrast, if we're talking about concentrations such as percentages, because that water is in the as-fed basis, it's less concentrated, meaning it'll be diluted. And when we take out the water and we're expressing nutrients on a dry basis, it's more concentrated. It would have a higher percentage of that nutrient because we've removed that water and we've removed that dilution. And so that will kind of come to sense a little bit more so in a week or two when we really focus on converting between nutrient expressions between dry matter and as fed. But understanding that basic concept helps us to understand some of our chemical analysis that we do for feeds. The first chemical analysis that we're going to talk about is an older system that was developed, I think, in the 1800s. And many of the elements are still used today. However, we have improved on some of them. But the initial development of the proximate analysis was to estimate the classes of nutrients. And when we talk about estimating, it's a very loose definition, and that's expressed in some of the names of these nutrient classes. The first that we talk about is moisture. It's one of the easiest things we can do. We can take a feed sample, weigh it as we get it, as it's an as-fed value, throw it in a drying oven for 48 to 72 hours, remove all the moisture, and figure out the percent moisture or the percent dry material in that feed. And that's going to kind of give us an idea of the water content, which is one of the 6 classes of nutrients. The next one we're going to look at is a crude protein. And it is called crude because it is a very loose estimation of the protein value of a feed. There's some different ways. We usually actually measure the nitrogen content and then calculate protein from that using some standard values. That's why it's called crude protein. The next one is taking a look at the lipids or fats, which is yet another nutrient group. And that's called either crude fat, or sometimes you'll see the actual procedure is called an ether extract. It's where you boil a sample in ether overnight, and that ether removes all the fat compounds from that feed, and you're able to estimate what percent fat is in that diet. The ash estimates the inorganic element or the total minerals. We have to be careful with ash because it's literally burning up a sample until there's nothing organic left in it and all you're left is with an ash residue. But it doesn't tell us the specific minerals. Ashing is actually a procedure that's generally used as an initial step to do more specific mineral analysis. The last two that we're going to talk about are the last two elements of the proximate analysis are crude fiber, which is a way of estimating the plant cell wall or that fiber content of a feed, and the nitrogen-free extract, which is calculated and is an estimate of the sugar or starchy carbohydrates of that feed. Again, these 6 categories, moisture, protein, fat, ash, crude fiber, and the nitrogen-free extract, are very loose definitions of those 6 classes of nutrients. If we take a look at the proximate analysis from a schematic standpoint, we're going to start off with a basic dry matter and drying down that feed to remove the water and get to an estimation of that dry material. Then we can run it down a couple different chains of event. The first would be what we call a Kjeldahl procedure, which involves boiling samples in acid, and it's not really fun to do. I've done a lot of them in my career. But it gets us to estimating the nitrogen content of that feed, from which we can roughly estimate a protein value so that we can work on developing our rations. Again, it's very crude though in a generalized estimation. The second procedure is actually taking and doing the ether extraction, boiling it in ether, removing the lipids. From there, we can actually boil in an acidic solution and in an alkaline solution following that, and we'll remove all of the starchy components of that feed and allows us to estimate that crude fiber. If we take that crude fiber then and burn it down, we're able to estimate the ash component of it and really separate out these different elements of a feedstuff to try to get a generalization of those 6 classes of nutrients that are so important from a feeding standpoint. However, that system is not perfect. What we found was that estimating the carbohydrates in the proximate analysis wasn't really accurate. There was a lot of crossover. It didn't separate it into as descriptive a categories, because some of the fiber would wind up in the nitrogen-free extract and some of the NFE would wind up in the fiber, and it wasn't as clear-cut as we'd like it. Some researchers at Cornell University in the 70s, Peter Van Soest, worked on developing the detergent fiber system, and especially dealing with forages and high-fiber feeds. That's what this system is developed for, and it's a more accurate measure of fiber. And there's 2 major components of this system, either a neutral detergent fiber or acid detergent fiber. Neutral detergent fiber contains the majority of the plant cell wall, the hemicellulose, cellulose, and lignin. It can vary in digestibility and is actually related to the bulk of a diet. Feeds high in neutral detergent fiber are going to be high in bulk density, very bulky, take up a lot of space, and fill up an animal faster. In contrast, the acid detergent fiber actually removes that hemicellulose, leaving us with the really low digestible cellulose and lignin, and acid detergent fiber is actually a way to estimate digestibility. Feeds high in acid detergent fiber are actually low in digestibility. So it provides us a way to look at the bulk and the digestibility of a forage from two fairly simple procedures that are easy to do in a lab. If we take a look at the schematic for the detergent fiber system, we're going to take a forage sample and run it through basically like a little washing machine with a neutral detergent. Think of like doing your laundry with Tide. It's a neutral pH, it's a gentle washing process, and what we do is we separate out the cell contents, which is considered very soluble and digestible, leaving us with that NDF fraction, the hemicellulose, cellulose, and lignin. Then if we were to take that remaining NDF fraction and run it through an acidic detergent, think of, again, your laundry but with a really low pH, and we're going to be able to wash out that hemicellulose, leaving us with the cellulose and lignin, and providing a way to estimate that really lowly digestible portion of a forage. The take-home message from these 3 videos involving nutrients is making sure we can define what nutrition is and what a nutrient is, talk and be able to name and describe the general functions of the 6 classes of nutrients, be able to define energy and discuss what measures of energy are used for various species and how energy is lost from the animal's body, define what the dry matter versus the as-fed basis is, and probably the air dry as well, and be able to define the elements of the proximate analysis and their key components, define the detergent fiber system and its key components. That's the essence of what you need to know from this series of videos.